Before the first keynote start, and thank you for all the attendees on site to attend this conference and Internet Summit, and thank you for the attendees online. And there's for this event, it's close to 1,000 experts registered listed this event. So thank you for your coming. And before the first keynote start, I would like to invite Professor陳宜君to the stage, who is currently Director of Infectious Disease Division at National Taiwan University, to introduce our first speaker. Let's welcome Professor陳宜君.
I'm Yi-Jun Chen. It's my honor to introduce our first keynote speaker, Professor Charles Y Chiu. He's professor of Department of Lab Medicine and Medicine, Division of Infectious Disease. Also, Associate Director of UCS Clinical Microbiology Lab, as well as a Director of UCS Arboviral Diagnostic and Discovery Center. His multiple functions represent an important role from bedside to benchside, from the clinical need to novel development. Why he had this unique role? Because his special background, he received a bachelor in electrical engineering and computer science, and a PhD program in biophysics. Later on, he received a full training in internal medicine and an infectious disease specialist. Today, he will give us a very special topic, metagenomic mNGS and host response profiling for diagnosis of infectious disease. As our chairman opening emphasized that the challenge of the diagnosis of etiology of infectious disease. In addition, infectious disease represent a prototype of precision medicine. We need to approach the etiology as well as the host response. Let's welcome Professor Chiu.
Thank you for that generous introduction. And I would like to start by thanking the organizers for inviting me to speak with you today. I'm going to talk about today metagenomic sequencing and host response profiling for diagnosis of infections. I do have a few disclosures. I will be discussing very briefly the software that we use to identify pathogens from next generation sequencing, which has been filed by my university as a patent application. And I'm also on the scientific advisory board and a co-founder for some companies related to NGS. I want to start with this slide because this slide really highlights the diagnostic challenges that we face in infectious diseases. It's known that in cases of pneumonia, anywhere from 15 to 60% of the time, we're unable to diagnose the cause of a respiratory infection in these patients, many of whom are in the intensive care unit. With fever and sepsis, about 20 to 40% of the time, the cause remains unknown, and this is despite extensive testing. And probably our most challenging infectious syndrome or potentially infectious syndrome is meningitis and encephalitis, where roughly about 50% of the time, despite extensive testing that's done in hospitalized patients, we're unable to make a diagnosis. And it's well known that the failure to obtain a timely diagnosis leads to delayed, inappropriate therapy, increased mortality, and excess healthcare costs. So our plan is to develop clinical metagenomic next-generation sequencing, otherwise abbreviated mNGS, as a technique that we can use to diagnose patients who may have acute infections. Ultimately, the goal is to bring it into the clinical laboratory, as we've done here in the U.S., and which is also being done in Taiwan. You can see here that the goal is to bring the research-based tool into the clinical lab, where we can prepare samples and do the sequencing in the clinical laboratory, analyze it using custom bioinformatics, and then produce a clinical result. Ultimately, the goal is to produce a clinical result that can be cost-effective and actionable, and can provide information for clinicians to treat patients that can hopefully result in improved patient outcomes and lower healthcare costs. Unlike other NGS applications in perhaps other fields, such as cancer or prenatal testing, genetic testing, with infectious diseases, turnaround time is really of the essence. you want to have your assay completed within hours to days rather than days to weeks, because otherwise your patient can quickly become worse and decompensate. And really, it's a very simple concept in that nearly all pathogens, because they're composed of DNA or RNA, in principle can be identified using a single sequencing test. The idea is to take a clinical sample to sequence all of the DNA and RNA in that sample, whether it's spinal fluid, blood, body fluids, or tissues. And then we can identify based on the proportion or the alignments to non-human reads, whether or not a patient's infected with a bacterium, a virus, a fungus, or a parasite. And we've demonstrated the clinical utility of this approach and its application to a variety of different body fluids in a number of publications that I show you here. And we currently are offering this diagnostic testing as a laboratory developed test, as a clinical test that's run from the UCSF Clinical Microbiology Laboratory. We run clinical assays from spinal fluid, from plasma, and from other body fluids using mNGS to identify infections. In addition to that, one important aspect of mNGS is that the ability to analyze the data. And we've developed this pipeline, which we call SERPI, which stands for Sequence-Based Ultra-Rapid Pathogen Identification. The goal of this approach is to directly address the computational analysis bottleneck to be able to take in clinical data from the sequencer to be able to upload it to the cloud. And by using cloud computing, we can analyze the sequence data for the presence of pathogens, whether they're viruses, bacteria, fungi, or parasites. What's generated is an automated results report and heat maps that can be reviewed by the laboratorian. This gives you kind of a look into the black box of what SERPI does. Essentially, what it does is it takes reads which are raw reads coming from the sequencer, pre-processes them, and then it aligns those reads to the human host. This is to get rid of human host reads, what we call computational background, host background subtraction. Later on, I'll be talking to you about how we're actually leveraging those reads to look at host response. But for the purposes of pathogen identification, we remove the human reads. The remaining reads are then aligned to all of the sequences or all of the microbial sequences in GenBank. And we implement some filtering and taxonomic classification algorithms that allow us to identify what pathogen is in the clinical sample. We've also established automated thresholds for what we would call a positive, such as a requirement for detection of an RNA virus is that there are reads mapping to 3 or more regions, or a reads per million ratio metric of greater than 10 for identification of a bacterium, fungus, or parasite. We've demonstrated the clinical utility of this approach in a paper that we published several years ago in the New England Journal of Medicine. The first author of this study is Dr. Michael Wilson, who will be speaking after me. I won't be talking about this particular study, but very briefly, it was a prospective study that was meant to compare the clinical utility of metagenomic sequencing, mNGS, against all other testing. This includes culture, PCR, serology, antibody, and antigen testing. And in short, the findings were that roughly a quarter of all diagnoses, infectious diagnoses, were made using metagenomic sequencing, including several diagnoses, the majority of which the diagnoses had direct clinical impact and were clinically actionable in terms of changing antibiotic regimen or affecting treatment. Since then, and this is unpublished data, you can see here that this is a representative set of all of the potential positive cases that we've identified over the years. And they include a wide variety of different pathogens of all types, including several pathogens which you may be unaware of or which are very rare or uncommon. They include cases of Potosi virus, astrovirus, in spinal fluid, Balamuthia mandrillaris, otherwise one of the brain-eating amoeba in spinal fluid, including several atypical uncommon bacteria and fungal species. The other thing that we've been able to show is that the technique doesn't merely apply to spinal fluid, but we can also do metagenomic sequencing of, in fact, a wide variety of other body fluids for infected patients. And we're essentially running the same assay on all of these body fluids to be able to identify pathogens that may be present in them. This has been published in a study in Nature Medicine. Since then, what we've been working on is we've been working on ways to optimize this assay. And the idea behind this approach is, can we actually simplify the actual assay protocol? There are a couple of goals for this. One is that we want the assay to be able to be run routinely in clinical labs which may not have mNGS expertise or may not have bioinformatics or complex testing expertise. The second reason is we want to be able to decrease and automate as much of the protocol as possible. And the third reason is we want to really decrease the turnaround times. Ideally, we want to be able to run this assay with same-day results. And this has been done for a respiratory assay that we've developed. This is specifically an assay for identifying respiratory pathogens, in fact, respiratory viruses. And it's using mNGS, so it can identify, for instance, novel viruses. This is done with the support of some US governmental agencies, including BARDA and the US Centers for Disease Control. But the goal of this is to develop an assay that could be implemented widely that would help us rapidly identify perhaps the cause of the next viral pandemic. And some of the improvements that we've made is we've moved, for instance, the library prep step to an automated instrument, which is essentially has 5 minutes hands-on time and can generate a library from either DNA or cDNA within 5 hours. We've also moved the sequencing to an Illumina mini-seq instrument in rapid run mode where we can do a run in 5 hours. We've implemented some improvements in the actual cDNA synthesis procedure as well as an extraction procedure such that those steps can be accomplished within 2 hours. And ultimately, we've decreased the test, which had a 2 to 3 day turnaround time down to 12 hours. We've also decreased the complexity of this assay because we now have a test that goes from 150 steps that needs to be run in the clinical lab down to 15 steps. I want to give you some idea of the performance of this rapid assay. And you can see here that the limits of detection are pretty good. This is using probit analysis for detection of these 4 respiratory viruses in respiratory secretions. You can see here that we're getting a limit of detection of about 500 copies per mil, which is comparable to many of the PCR-based assays that are available today. I also want to demonstrate some of the clinical use cases or applications of this technology. So one thing that can be done, for instance, is that we can use the reads to rapidly identify or genotype the particular variant that may be infecting a patient, the infecting variant in patients who have COVID-19, who are infected with SARS coronavirus 2. That's what you can see in the upper left. In the lower left, you can see where we can identify, for instance, very unusual viral infections that are typically not looked for with common assays that are done in the hospital. And importantly, all of these infections are actually from patients who have critical illness. They have a critical pneumonia because of this, and many of whom will remain undiagnosed for several days. You can also see on the right, we can identify more pathogenic rhinovirus and enterovirus strains. In particular, there's been a lot of concern with regards to enterovirus D68 and enterovirus A71, both in the US as well as in Taiwan, and its association with this severe clinical syndrome called acute flaccid myelitis. And you can see here that using mNGS, we can not only identify the presence of an enterovirus or rhinovirus, but we can also rapidly subtype it. I also want to show you kind of an application of this technique. A clear application is, can this technique be used to identifying the next cause, the next viral pandemic? And these are, some of the viruses that we've been worried about over the past decade. But, how are we going to be able to ensure that we can identify the next pandemic virus, especially if it happens to be a very divergent virus? We know that these are RNA viruses typically that may mutate constantly. And there's a possibility that by using kind of routine primers and probes, we might not be able to identify it. So here's where I performed an in silico demonstration. And the idea behind this is we generated in silico essentially a synthetic data set. What we did is we took every single viral genome from the database, We then cut it up into non-overlapping segments. And the idea is we took those segments and we spiked them into negative reads from a human background matrix library. And then this is kind of the test. We actually ran it through our analysis protocol, but we ran it comparing it to a reference database that did not have any human viral sequences. So the idea is if there's no sequence to influenza viruses, if there's no sequence to Ebola, no sequence to SARS coronavirus 2 or any other coronaviruses, can the algorithm potentially identify a novel virus based on homology to either non-human, plant, or animal viruses? And the answer here is actually yes. This is basically a list of all of the common or well-known pandemic-causing or outbreak-causing viruses that we're aware of that cause human disease and human outbreaks. They include, for instance, hantaviruses, Ebola virus, Mpox or monkeypox virus, SARS coronavirus 2 and 1 and other coronaviruses, other neurologic arboviruses, including West Nile virus, West equine encephalitis virus, and Venezuelan equine encephalitis virus. And the idea here is that we can still identify the virus even if we have no reference in the database, and that's because there is a relative, either an animal virus or a plant virus. And we are indeed well aware that most of these viruses, in fact, nearly all of these viruses are actually zoonotic. They come from viruses that have cross species and now kind of infect the human population. And so one particular virus I want to actually highlight here is chikungunya virus. Chikungunya virus is a member of the Togaviridae family. It's actually the only human virus representative, and in fact, the only representative of the Toga virus or the Togaviridae family. And in fact, the closest relative to chikungunya virus actually happens to be a plant virus in the alpha flexiviridae family called vanilla latent virus. This is a virus that infects the vanilla plant. But you can see here that we can actually identify chikungunya virus despite having no chikungunya virus sequence in the database by virtue of its homology or similarity to vanilla latent virus. And this is where we spike it in silico at levels as low as 10 reads per million. So this really suggests that should we implement metagenomic sequencing, we will indeed be prepared for the next pandemic virus, no matter what it happens to be. I want to give you some example because we are discovering these, quote, novel viruses all of the time. This is actually a case of Potosi virus meningoencephalitis. Now, probably the majority of you have never heard of Potosi virus, and that's because this is not a human virus. This is a virus that actually sequenced from mosquitoes in Connecticut in the United States about 20 years ago. And since then, it has never been described, it's never been detected before in humans. But we identified this virus actually in spinal fluid from a 70-year-old gentleman who lived in the rural Midwest and actually had been bitten hundreds of times with mosquitoes. And so this is an example of another potentially emerging virus. This is yet another example. This is a 60-year-old gentleman who had common variable immunodeficiency, so an immunocompromised patient who is an avid hiker. He was actually a hiker from Idaho in the US, was admitted with encephalopathy. And we found in spinal fluid reads using metagenomic sequencing a virus which had about 70 to 90% nucleotide homology to Lone Star virus, which was a virus that was found in the Amblyomma americanum tick. But this is another virus which has never been described as causing human infection. So I think these two examples are really highlight kind of the importance of metagenomic sequencing as potentially a monitoring or surveillance tool that we would be able to use to identify the next virus that may be the cause of an outbreak or maybe the cause of these unexplained infections. Unfortunately, both of these patients died. There really isn't a treatment for these types of infections, and they both presented with very severe disease. But the idea is that, possibly if the diagnosis had been made earlier, for instance, for this patient, some potential antiviral treatments, experimental antiviral therapies that are not approved, such as ribavirin or intravenous immunoglobulin, might have been considered. I want to move also into some really exciting area of work and the idea here is we want to be able to leverage metagenomic sequencing as a way to make indirect diagnosis of infection. So the idea behind this is traditionally in microbiology, we diagnose infections by identifying the pathogen. We either grow up the pathogen in culture, We either do antigen testing to look for proteins from the pathogen or the virus. We do nucleic acid testing or PCR to amplify DNA or RNA, or we can do metagenomic sequencing, which is what we're talking about here. We have very limited tests, however, to be able to diagnose infections on the basis of the host response. One example is serology, where we can identify, for instance, antibody or a serologic response that may give us some clue as to what is causing the infection. There are some disadvantages, of course, to serology. One is that it can take several weeks for patients to mount a serologic response. Immunocompromised patients may not have a serologic response at all, and serology can be non-specific as well as insensitive. We also have other metrics, and this was actually discussed by one of the introductory speakers. We have ways of measuring the cell counts in spinal fluid or glucose or protein levels, et cetera. We can use these as potential biomarkers, but the major disadvantage of this is that these tend to be very non-specific. I mean, we can see lymphocytosis, for instance, elevated lymphocytes in spinal fluid, but it may correspond to a viral infection, a fungal infection, or even an atypical bacterial infection. We also have some potential biomarkers that can assay protein expression, host protein expression, such as C-reactive protein or CRP or procalcitonin. But again, one of the major drawbacks of this is that these responses can be non-specific. And so what we want to do here is why not leverage the human reads that we're actually getting from metagenomic sequencing. So this is the concept here. And this is actually one of the goals of what we call precision medicine. And the idea behind precision medicine is either by sequencing a patient's cancer genome, by sequencing a patient's genome to look for rare diseases, or sequencing a patient's transcriptome to look for a host immune response to infection, can we more accurately diagnose diseases in these patients? What makes this quite exciting is, in principle, we can not only potentially identify infectious diseases, but potentially even non-infectious diseases such as autoimmune disease or autoantibody syndrome, which I think Dr. Wilson will be talking more about this, about kind of the differential diagnosis of patients with encephalitis and meningitis. In addition, it brings up the concept that we might be able to also diagnose acute and chronic disease. It's much more difficult to diagnose chronic infectious diseases because the DNA from the pathogen or RNA from the pathogen or protein may be gone by the time you actually collect the sample in chronic disease. And so the general idea is this. Imagine that if we do the same metagenomic sequencing, but what we're doing here is we're using the non-human reads to identify pathogens, but we're using the human reads or the RNA reads to identify signatures that can help us identify the host response to the infection. And so this is done with a traditional machine learning analysis workflow where we take lots of samples. 80% of the samples, in fact, are used to develop a model that's called a training set. And then we apply the model to a test set of 20% of the samples, which are independent. And the idea is, based on the performance of the test set, we can derive metrics as to how accurate is the model, what's the sensitivity and specificity of the model. And in fact, this has been done from spinal fluid from our CSF metagenomic sequencing. Because, as I said, we've been running this test for over 5 years, we now have access to thousands of samples. So what we can do is we can take those non-human reads, we align them to identify pathogens, but we can now take the human RNA reads and assay the host response. And this is done with the help of a bioinformaticist in my lab, Charlie Omura. But this is another way to look at it. It's rather complex, but I just want to focus on the overall structure of this. So the idea is, imagine we're taking a wide variety of different samples, some from patients with infections, bacterial, fungal, viral, autoimmune, and some patients who may not even have any infections, may have a non-infectious cause like autoimmune disease or lupus or amyloidosis, et cetera. And the idea is to develop a training set and generate a model that can help predict or classify what kind of infection or non-infectious disease the patient has. And so ultimately what we've done is we can select genes that can help us distinguish one category from another category. So for instance, to give you some idea, this is the set of genes, the top genes that can distinguish bacterial infection from everything else. And as you might expect, many of the genes that you can see on the right are associated with the bacterial host response. In fact, the top gene that we can see here is PI3, which is an antimicrobial peptide specifically generated by the host in response to bacterial infections, along with macrophage-associated responses. With viral infections, as you can see on the lower right, there's a strong association with interferon-associated genes or cytokines. And with fungal infection, we also see genes which have been implicated in fungal disease. With autoimmune or non-infectious syndromes, as you might expect, we see genes that have been linked to cancer, to neurodegenerative disease, or to autoimmune disease. And these are indeed all of the non-infectious categories that can cause a meningitis or encephalitis. But ultimately, the proof is in the pudding. How good is this classifier really? So when we actually apply this classifier in principle and we developed an algorithm that you can see here, we're actually getting pretty good performance, anywhere from 81 to 87% sensitivity and 93 to 95% specificity. When we combine everything, you can see here that we're getting an overall accuracy in kind of around 83% across the test set. And so this is basically where the model is accurate enough that you can envision providing this information. One of the benefits of this is that we're not generating any new data. We don't have to do any new work. We're simply repurposing the data, reanalyzing it to provide host response information. And so what we've done next is we decided to go through 1,150 consecutive patients at University of California, San Francisco. These are patients who all have an unknown meningitis and or meningoencephalitis. Approximately 400 of them have an etiology of illness that was clearly identified. And so those samples, they underwent blinded chart review. What we needed to do is the problem with comparing this to evaluate the performance of the model is we don't have a gold standard. As I said, very often we never get a diagnosis. So the idea here is, can we basically classify our confidence that the diagnosis that was made clinically is true? And so the confidence score varies from 1 to 4, where 4 means that there's very strong evidence that we're very confident that the clinical diagnosis was correct, whereas 1 means that was low confidence, where we have kind of no laboratory confirmation and we're less confident that the clinical diagnosis is correct. And what you can see is, which makes sense, the performance of our host response classifier is highly dependent on the confidence of the samples that we're using to evaluate the classifier. So if the confidence score is very high, you can say the classifier performs very, very well. But the accuracy of the classifier goes down if you're including samples where we don't have a gold standard diagnosis. I also want to say, though, and this makes me quite excited, is the fact that we can now do subcategory classification. And the idea behind this is that we can use a leave one out algorithm to be able to not only identify whether an infection is viral, bacterial, or fungal, but can we, in fact, even identify what type of infection it is? And in particular, for instance, can we use this to identify Mycobacterium tuberculosis, which is very difficult to diagnose in the setting of meningoencephalitis, and which culture can take weeks. PCR is insensitive, and we found that we can actually identify mycobacterial disease or cocci plus histoplasma. These are dimorphic fungal infections, parasitic infections, which are also difficult or challenging to diagnose with relatively good performance. This gives you an example. We can identify, for instance, acute phasamyelitis that's associated with either enterovirus A71 or D68. And what makes this approach particularly powerful is the fact that we cannot identify D68 or A71 directly from spinal fluid, either using PCR or culture. This is where we cannot identify the pathogen, and we have to resort to host response. I want to actually also go into this other area, which is now we're hoping to actually be able to identify non-infectious syndromes that may mimic an infection. These include, for instance, acute cases of neurosarcoidosis, autoantibody syndromes, cancer, lupus, multiple sclerosis, or other vasculitis or other non-infectious etiologies of meningitis and or encephalitis. So this is very preliminary work, but ultimately I'm hoping that we can identify the full spectrum of infections that cause disease. I want to end by providing perhaps a few examples, 3 examples of how we've directly applied this metagenomic sequencing to address outbreaks. Dr. Michael Wilson will be talking about case reports or specific clinical cases. I'll be focusing here on outbreaks. So we reported in the journal Nature this year using metagenomic sequencing to identify adeno-associated virus 2, which has never been really linked to any disease, in association with cases of severe acute hepatitis in children. Throughout the United States, and in fact, throughout the world, there have been cases of severe acute hepatitis of unknown etiology with extensive workup. And this happened as children were starting to go back to school after the COVID pandemic. We were able to identify a large number of viruses in these clinical samples. In fact, on average, these children were infected from blood with at least 3, and in some cases 4 viral infections with either adenoviruses or herpes viruses. But what was quite interesting is that we also identified adeno-associated virus 2, and this is basically a virus that actually it does infect liver cells and infects the liver, but it's never been associated with disease. In fact, AAV is actually commonly used as a gene therapy vector, but we have found that it was not only AAV, but one specific strain of AAV called AAV2, which has been linked to severe cases of acute hepatitis. And then when we do sequence analysis of this, the other thing that's quite interesting is that independently, 2 groups in the UK, in England, actually discovered the same strain of AAV2 in association with cases of acute severe hepatitis in children from England. So really around the world, in 2 disparate areas, geographic areas around the world, we've actually found the same strain. And there's also been some evidence to suggest that this may also be linked to a specific HLA type or a specific genetic predisposition for these children to develop severe hepatitis. I want to talk about a second example. This is actually a fungal meningitis outbreak that was associated with procedures performed under anesthesia in Matamoros, Mexico. These are patients who they crossed the border from the US to Mexico to get some cosmetic surgery, plastic surgery procedures. Unfortunately, from all of the patients, or a large number of patients from one particular clinic, came back and developed a meningitis and encephalitis, of which there were, to date, there are about 5 fatal cases, unfortunately. And so we identified, and this is in collaboration with the CDC, a specific case, again, using metagenomic sequencing of spinal fluid, a case of fusarium, actually as the possible cause or likely cause of this fungal meningitis outbreak, Fusarium solani. And in fact, we identified it, and this is from one patient where you can see we had metagenomic readings that actually spanned most of the chromosomes most of the chromosomes corresponding to this organism. But one of the key questions that we want to introduce here is, are these linked? In other words, are these infections all caused by the same strain of Fusarium? So we can actually harness metagenomic sequencing, and what we can do here is we can actually develop a technique by using the metagenomic reads that align to Fusarium. We can actually generate, we can do a multiple sequence alignment and generate essentially a phylogeny. And we've been able to show by generating a phylogeny that all of those strains were actually associated with the same, where they were all the same strain, which suggests that there was a common source. It's now thought that it was an anesthetic drug that was contaminated with this fungus that actually resulted in the infection. And I want to add, but with one last example, this is a transplant-associated outbreak. It was a chance, basically, this is an outbreak of a putative infection where the tissue organ donor actually spread the disease to 5 recipients because through the organs that were donated, unfortunately, 2 of those patients died. And so the question is, what is this? Is this bacterial, viral, fungal, parasitic, even potentially prion-associated? And we were able to identify from a single read out of 15 million that this was a case of yellow fever virus infection that actually came because the blood donor who had actually received the live yellow fever virus vaccine had donated to the tissue organ donor, and the tissue organ donor had unfortunately donated infected organs to these recipients. What's actually quite remarkable is PCR testing of spinal fluid from all of the recipients was negative. In fact, the diagnosis had to be made, unfortunately, or the confirmation of the diagnosis had to be made by brain on autopsy, where it was finally positive by RT-PCR. But one thing I want to highlight here is this is where looking at the host response can give you some clues, because if you look at all of the recipients who actually had severe disease, all of them had a very high probability that this was a viral infection. And what you see on the right, those particular genes are associated specifically with yellow fever vaccination or yellow fever infection. And so this gives you another potential application of using host response to enhance your metagenomic sequencing. I would like to end by thanking all of the members of my lab who did this work, funding from several organizations, as well as collaborators from the CDC, from UCSF, and from other institutions. Thank you very much, and I would be happy to answer any questions. Thank you.
Thank you, Professor Chiu, for your splendid talk. I believe that many audience want to have a lot of ideas to communicate with you, but please save your questions until panel discussion session.